Imagine, for a second, walking into this massive state-of-the-art fire station.
Okay, I'm picturing it.
Right, so the garage doors are wide open, the heavy red fire trucks are just sitting there in the bays, and the engines are absolutely roaring, like shaking the floorboards.
Wow, so they're totally ready to go.
Exactly. The firefighters are fully geared up. They've got the heavy coats on, helmets strapped, hands are literally gripping the steering wheel, staring intently through the windshields.
Right, just waiting for the call.
But you look around and there's no fire. You step outside, no smoke in the sky, no alarms are ringing anywhere.
That's eerie.
It is. In fact, this particular station has been quiet for decades. So why on earth are they prepped and waiting, burning fuel, ready to burst out the doors at a millisecond's notice?
Well, it's a surreal image, but it actually beautifully captures the profound biological mystery we are unpacking today.
It really does.
Because in biology, we typically expect ruthless efficiency, right? I mean, if a cell isn't actively using a set of instructions, it should pack them away. It should shut the systems down to conserve energy.
Yeah, nature doesn't like to waste energy.
Exactly. But sometimes, as it turns out, the cell just keeps the engines running anyway.
Okay, well, let's unpack this. Welcome to the deep dive, everyone. Today, we are exploring this just monumental 2026 paper published in Nature Neuroscience.
It really is a landmark paper.
Yeah, spearheaded by Mukunkabe, Gonzalo Castelo-Branco, and a massive international team. And our mission today is to explore their discovery of something called epigenetic memory, specifically in the adult human central nervous system.
Okay.
We're going to look at how your adult brain and spinal cord cells secretly remember their own fetal development, why they keep certain biological engines idling for your entire life.
Which is just wild to think about.
Totally wild. And how this bizarre phenomenon holds the key to both, miraculous spinal cord regeneration and, on the flip side, some of the most devastating brain tumors known to medicine.
So to really grasp the scale of what this team accomplished, we have to kind of distinguish between the genome and the epigenome.
Right. The basics.
Yeah, most of us know the genome. It's the DNA sequence, right? The biological hardware. And that hardware is basically identical in every single cell of your body.
Whether it's a skin cell or a heart muscle cell.
Or a neuron, exactly. But the epigenome, that is the software, it's this incredibly complex regulatory layer sitting on top of the DNA that dictates exactly which genes are turned on and which are firmly turned off.
Right, because a brain cell and a liver cell have the exact same instruction manual. They just have completely different chapters open.
That's a perfect way to put it. And, analyzing that software in human tissue is incredibly difficult.
I can imagine. But this team, they looked at postmortem central nervous system tissue from 20 human donors, right? Ranging in age from 34 to 74.
They did. And they analyzed 3 distinct regions: the primary motor cortex up in the brain, the cerebellum, and the cervical spinal cord.
And they did this all at the single-nucleus level, examining literally tens of thousands of individual cells.
Which is a staggering technical achievement, especially with frozen archival tissue.
Yeah.
But before we can actually read this cellular memory they found, we have to understand the tools they use to find it.
Yeah, the technology here is like half the magic of the discovery. Let's get into the how of this study. The first major tool they used is called SNATAC-seq.
Right, single-nucleus assay for transposase-accessible chromatin.
Which is a mouthful. But I love the analogy of the genome as a massive, incredibly dense library containing every instruction manual for your body.
Oh, for sure.
Because there is so much information, most of the books are locked away in dark, totally inaccessible vaults. So SNATAC-seq is essentially the tool that maps the physical architecture of that library.
Yes.
It tells us exactly which doors are physically unlocked and pushed open, exposing the DNA so it can potentially be read.
That's a perfect way to visualize it. Inside the nucleus, your DNA is wrapped around proteins called histones, which forms this complex called chromatin. When chromatin is tightly packed, the door is locked. When it opens up, the SNATAC-seq tool uses this hyperactive enzyme to slip into those open regions and just drop a tag.
So that gives us our map of the open doors. But, I have to push back a little here. Just knowing a door is open isn't quite enough, is it?
What do you mean?
Like if I walk down a hotel hallway and see a door propped open, it doesn't automatically mean someone is inside watching TV. It just means the room is accessible.
Ah, exactly. An open door doesn't mean anyone is actually inside reading the book.
Right. Which is why the team had to use a second super cutting-edge tool called nano-CATion tag. They adapted this to look at the specific chemical tags left on those histone proteins you just mentioned.
Precisely. They were looking for two specific marks, and they mapped them simultaneously across the entire genome. The first is called H3K27AC.
Okay, let's simplify that one for memory's sake. We'll just call it the action mark.
Action mark. I like that.
Yeah, the AC in the name stands for acetylation. But if you think of it as action, it makes perfect sense. It's a green light. It marks regions of the DNA that are actively being transcribed and used by the cell.
Spot on. And the second mark they look for is H3K27E3.
And we can think of that one as the stop mark or maybe meltdown. The ME3 is trimethylation, but functionally it's a red light.
Right. It marks regions that are tightly repressed, shut down, and just locked away.
So by combining these tools, the researchers weren't just guessing what the cells were doing.
No, not at all. They could see exactly where the DNA was physically unwound and accessible, and then they could look at the functional state of that open region.
Right, like was it marked with a green action tag, meaning workers were inside reading the instructions, or was it covered in red stop tags?
Exactly. So let's do a quick recap here. We are mapping the brain using SNATAC-SEC to find the open doors and nano-CT and TAG to find the action versus stop signals.
Which brings up a fun question for you listening. If you could look at the open doors in your own cellular library right now, what forgotten instructions do you think are just sitting there?
It's a great question to keep in mind as we move forward, because now that we have this incredible toolkit, the logical next step for Kabi's team was to point this tech at the actual hardware of the nervous system and map the different cell types.
Right, and they mapped all the major players. They looked at excitatory and inhibitory neurons. They mapped microglia, which are like the immune sentinels of the brain.
They mapped astrocytes too, the crucial support cells. But the absolute stars of this deep dive turned out to be the oligodendrocyte lineage.
Yes, the OLGs. For anyone who isn't super familiar, oligodendrocytes are basically the unsung heroes of the nervous system.
They really are.
Their primary job is to create myelin, and myelin is the fatty insulating sheath that tightly wraps around the long cables of your neurons.
Think of like the rubber insulation on a copper electrical wire.
Exactly. If you don't have that insulation, the electrical signals in your brain and spinal cord degrade, they leak, or they just short circuit completely.
They are entirely essential for human life. So the researchers specifically focused on both the mature, fully functioning myelinating cells and their parents, the oligodendrocyte precursor cells.
Or OPCs, right, which are essentially these stem cell-like reserves floating around in the brain.
Yep, exactly. And while mapping the action and stop tags in these specific insulating cells, the team hit a gold mine. They discovered a brand new distal enhancer for a gene called SOX10.
Now, SOX10 is famous in neurobiology. It's a master regulator gene. It is the absolute boss that tells a generic cell, hey, you are going to become an oligodendrocyte and you are going to survive and make myelin.
And to understand why finding an enhancer for this gene is such a big deal, you really have to picture how gene regulation works in 3D space.
Okay, paint a picture for us.
So if the SOX10 gene itself is a speaker, an enhancer is the volume knob. And what's fascinating about distal enhancers is that they are located super far away from the gene they control on the linear strand of DNA.
Wait, so how do they connect?
The DNA actually has to physically loop back on itself, fold up in three-dimensional space, so that the volume knob can physically touch the speaker.
Oh, wow. So finding a new volume knob hidden in the DNA that controls the most important gene for brain insulation is incredible. But, I mean, in modern biology, you don't just get to point at a map and say, I think this does something.
No, definitely not. You have to prove it.
And the way they proved it is straight out of science fiction. They use a technology called CRISPR activation, or CRISPR.
Right, so most of us are familiar with standard CRISPR, the genetic scissors that can cut and splice DNA. But here, the researchers used a modified, quote-unquote, dead version of the Cas9 protein.
Which they call dCas9.
Exactly. Its cutting blades have essentially been dulled. It can't cut DNA anymore.
So instead of a pair of scissors, it becomes like a highly programmable microscopic drone.
That's it. It's a GPS-guided drone.
Yeah.
And they loaded this drone with a really heavy-duty payload, a powerful transcription activator protein called P300.
Basically, they built a drone carrying a massive turn-on switch.
Yeah, exactly. And they programmed the coordinates for this newly discovered SOX10 volume knob into the drone, and they unleashed it into living, stem cell-derived human oligodendrocyte precursors.
They wanted to see, hey, if we artificially crank this specific enhancer that we just found, what actually happens to the cell?
And the results were beautifully definitive. When the drone landed on the enhancer and flipped the switch, The expression of the SOX10 gene just absolutely skyrocketed.
But crucially, the other neighboring genes on the DNA strand genes that just happened to live nearby but weren't part of this specific circuit, they didn't change their expression at all, right?
Not at all. The drone only triggered the exact target it was meant to. It was absolute proof of cause and effect. They found the secret physical control switch for the cells that insulate our neural wiring.
That is just a massive win for basic science.
It really is.
So let's do another quick recap. The study mapped major brain cells and discovered this hidden volume knob controlling the vital SOX10 gene, and they proved it using this crazy CRISPR drone technology.
Right.
So here's a question for you listening. Since enhancers can be located so far away from the genes they control, how do you think your body knows which volume knob connects to which speaker without getting wires crossed?
It's a huge puzzle. But, as the researchers kept poring over the epigenomic maps of these adult cells, moving from the brain down into the cervical spinal cord, they found something that didn't make any sense at all.
Yeah, this is where it gets really interesting. They found a ghost in the cellular machine.
It really did.
Because finding the SOX10 enhancer was, normal expected biology.
Yeah.
But when they looked specifically at the mature oligodendrocytes in the adult spinal cord, they found a pattern of open doors and action tags that simply shouldn't exist in a healthy 74-year-old human.
No, they shouldn't. They found massive anomalies in the HOX genes.
So let's talk about the HOX genes. They are legendary in developmental biology. They are basically the master architects of embryonic development.
Exactly. When you were just a tiny developing embryo in the womb, it was your HOX genes that laid out your fundamental head-to-tail body plan.
They are the instructions that make sure your arms grow where your arms belong. And, your spinal cord forms exactly the way it should.
They're the ultimate fetal instruction manuals. And because they are so incredibly powerful and potentially chaotic once you are a fully formed adult, those HOX genes are supposed to be locked away in the deepest, darkest vault of the cellular library.
Right. They should be completely covered in those red stop marks we talked about. They should be shut down tight.
But in the adult spinal cord cells, they weren't. The researchers found that the HOXA and HOXD gene clusters were just wide open.
The chromatin was highly accessible.
Yes, and the pattern of the action and stop marks was breathtakingly specific. It displayed this phenomenon known in biology as collinearity.
Okay, let's visualize this collinearity because it's super vital to understand. Picture an ancient, incredibly long instruction scroll. Okay. This scroll contains all the sequential instructions for building a fetal spine. What the researchers saw was that the first half of the scroll was completely unrolled, highly legible, and covered in those green action tags.
Right.
But then, right in the middle of the scroll, there is this sharp, abrupt transition. The second half of the scroll is tightly rolled up, glued shut, and covered exclusively in red stop tags.
And that exact spatial half-and-half pattern, the open active front and the completely repressed back, that is not a random mistake.
No.
It is the precise canonical hallmark of early fetal developmental biology. It is the exact epigenomic signature of a cell that is actively building a brand new spinal cord in a developing embryo.
Which is insane, because they were looking at it preserved perfectly in the spinal cord of an adult who had been fully grown for decades.
Exactly.
But wait, I'm stuck on something here. This is the decoupling paradox that the paper highlights, right?
Yes, it is.
Because if the front half of the scroll is unrolled, the doors are open, and the green action lights are all turned on, why isn't the cell actually building fetal tissue right now? Like, what's stopping the engine from shifting into drive?
That is the defining question of the whole study. Because when the researchers measured the actual RNA expression meaning, they checked to see if the cell was actively reading the instructions and actually manufacturing the proteins to build new tissue. They found almost zero activity.
Nothing.
Basically nothing. The DNA doors are open, the green lights are on, the transcription machinery is totally poised, but the workers aren't actually building anything.
It brings us right back to our opening hook. The fire station is open, the trucks are running, the drivers are at the wheel, but there is no fire, and they just aren't moving.
Right. Why are these genes idling? This is the epigenetic memory.
Okay.
The adult cell physically remembers its fetal state. It maintains the architectural blueprint of its own creation. It keeps the developmental engines primed and running, but without ever fully committing to producing the final protein product.
It really makes you wonder, though. Nature is notoriously ruthless. Evolution does not tolerate wasted energy at all. So what possible advantage could there be to keeping a cellular engine idling for 70 years without ever putting it in drive?
I mean, it's the multi-million-dollar question in neurobiology right now. Let's do a mini recap before we dive into that.
Good idea.
So adult spinal cord cells maintain fetal HOX gene patterns. The DNA is open and marked for action, but strangely, no RNA is being produced.
Which leads to a question for you all listening. Nature is deeply efficient. What evolutionary advantage could there possibly be to keeping an engine running for decades if you weren't going anywhere?
Well, we are transitioning here from the mystery of what is happening in these cells to the profound clinical implications of why it matters. The researchers proposed that this idling epigenetic memory basically acts as a double-edged sword.
Okay, let's start with the light side of the sword, which is regeneration. We all know the central nervous system is notoriously bad at healing itself. If you sever a spinal cord, the paralysis is usually permanent because the cells just can't bridge the gap.
Exactly.
But maybe keeping these developmental HOX genes primed and idling is the body's ultimate contingency plan.
That's the theory. If there's a catastrophic injury to the spinal cord, those cells might need to rapidly reactivate their ancient fetal tissue-building programs to attempt emergency repairs.
And if the library doors were locked and the books were buried under piles of red stop tags, it would just take far too long to mobilize a response.
The tissue would die long before the cell could figure it out. By keeping the epigenetic memory primed, the book is already open on the desk.
It's a beautiful, incredibly hopeful hypothesis. It suggests that our spinal cords might have this latent competence for regeneration, just waiting for the right trigger to, push the gas pedal.
It really is hopeful, but unfortunately, every biological backdoor that is left open can be exploited. And that brings us to the dark side of epigenetic memory. Gliomagenesis, or the creation of deadly brain tumors.
To investigate this dark side, the researchers had to go way beyond just looking at a linear map of action and stop tags, right?
They did. They used an incredibly advanced technology called micro-C to look at how the DNA physically folds in three-dimensional space inside the nucleus.
We touched on this earlier with the volume knob enhancer, but it's hard to overstate how complex this is. If you took the DNA out of a single microscopic cell and stretched it out, it would be about 2 meters long.
Which is huge.
Huge. To fit inside the microscopic nucleus, it has to fold into unbelievably complex 3D origami structures. Regions of DNA that are millions of letters apart on the linear strand are brought together in 3D space to interact and form functional loops.
And using high-resolution micro-C, the team actually mapped the exact 3D origami architecture of these idling HOX genes in normal, healthy human cells. They mapped the loops, the physical contacts, everything.
And the clinical connection they made from that 3D map is absolutely chilling.
Truly chilling.
When they compared the 3D folded architecture of these primed, idling HOX genes in healthy adult cells, it bore a terrifying, near-perfect resemblance to the chromatin architecture found in high-grade pontine gliomas.
Yeah, these are specific tumors carrying a mutation known as H3.3K27M. They are incredibly aggressive, deeply devastating brain tumors.
They're frequently found in the midline structures of the brain and spinal cord, often in children and young adults, sadly.
And what the researchers discovered by comparing these 3D maps is a profound and terrifying mechanism of cancer biology.
Because we often think of cancer as this incredibly inventive, chaotic monster that constantly mutates to find new ways to grow. But this research suggests something totally different.
Right. The tumor doesn't have to start from scratch. It doesn't have to invent a brand new, complex genetic pathway to grow uncontrollably.
It doesn't have to invent the weapon because the weapon is already lying on the table. The tumor simply hijacks the idling fire engine.
Exactly. If we connect this to the bigger picture of oncology, that specific tumor mutation essentially just acts as a heavy foot slamming down on the gas pedal.
Wow.
But the engine was already prepped, fueled, and running because of the healthy cell's fetal epigenetic memory.
The tumor just exploits the natural primed state of the HOX cluster to initiate massive, unchecked developmental growth in an adult body.
It is a profound realization.
It really is. Our vulnerabilities to some of the most aggressive, untreatable cancers are literally written into the historical, physical memories of our healthy cells.
It completely shifts our perspective. It forces us to entirely reevaluate our therapeutic targets.
Because when we treat these specific gliomas, we aren't just fighting a random foreign genetic malfunction.
No, we are fighting our own developmental history, weaponized against us.
Let's quickly recap that, because it's heavy. Epigenetic memory keeps cells primed for rapid regeneration, but this open state also provides a backdoor for high-grade gliomas to hijack developmental growth programs.
Which leaves us with a critical question for the listeners. If a tumor is basically just a confused cell trying to execute a fetal growth program in an adult body, How does this change the way we should think about treating cancer?
It's wild to think about. So what does this all mean? If we synthesize this entire journey from discovering new volume knobs to ancient fetal scrolls, we arrive at a powerful, almost philosophical realization.
I really do.
Your body is not a static machine. It is a living, breathing archive. Right now, every single glial cell in your spinal cord carries the silent, physical memory of the exact days you were a developing embryo in the womb.
And that memory isn't an abstract concept. It is physically, tangibly encoded in the 3D folding of your DNA and the chemical tags sitting on your histones. It means our biological past is intimately our biological prologue.
And the stakes of understanding that prologue couldn't be higher, because the very mechanisms that might one day allow us to heal paralyzing spinal cord injuries by figuring out how to safely put those idling engines into drive are the exact same mechanisms we must learn to outsmart and shut down in order to cure devastating brain cancers.
The duality of it is just staggering. The same open door offers salvation or destruction depending entirely on who walks through it.
Which brings us to a final provocative thought for you to turn over in your mind today. We've seen how researchers can now use CRISPR tools, like that microscopic drone carrying an activation switch to target specific enhancers and turn up the volume on genes.
Right.
We also now possess a high-resolution map of exactly what a primed epigenetic memory looks like in a healthy cell versus a hijacked memory in a tumor cell.
So imagine the future of medicine a few decades from now. Could we one day design an epigenetic therapy that selectively targets a brain tumor, sneaks into the library, and forcibly wipes its hijacked fetal memory?
Like causing the cell to instantly forget how to be cancer and revert to a normal state?
Exactly. Or flip that entirely on its head. Could we artificially induce this exact state of fetal epigenetic memory in other parts of the body?
Oh, wow.
Could we teach a failing, scarred adult heart or a cirrhotic liver how to act exactly like a fetal organ, allowing it to seamlessly regenerate itself from massive damage?
The epigenome is the ultimate operating system of life. The library of you is not just open for reading anymore, it is waiting for us to learn how to write in it.
It's an exciting frontier.
And as we learn to write, we have to remember, sometimes it's incredibly dangerous to leave the fire trucks running, but you never know when you might desperately need them to save the station. Keep diving deep, and we'll catch you next time.